there, welcome to lecture 3. This is Catherine Creech, and that's my adorable kitty cat, Callie. And my email's on the bottom. At any time, you're always welcome to email me. So let's go ahead and get started. Our question for today is, how does a cell make a protein? And the answer is there's actually a couple different ways, and it's a little bit complicated, but we'll go through it together. And hopefully you feel good about it at the end of the lecture. Here is our fabulous cell that we're going to use to make a protein. I know that this is a eukaryotic cell because this structure here, this big purple thing, is a nucleus. So nuclei are one of the major differences between prokaryotic cells and eukaryotic cells. Prokaryotes, pro being before, they evolved before the nucleus did, so they do not have nuclei. Then we have all these other really cool components inside the cell. We have these mitochondria, we've got the Golgi Apparatus, we've got the Endoplasmic Reticulum. Prokaryotes are also lacking those items because they do not have membrane-bound organelles. Only eukaryotic organisms have membrane-bound organelles. And all of this stuff is made out of membrane. Membrane is a phospholipid bilayer which has cholesterol embedded in it and carbohydrates and proteins, and it's really cool. It's got that fluid mosaic model of movement that you just read about for your homework. If you haven't done your homework yet, I highly recommend reading that before listening to this lecture because I'm going to have some assumed knowledge for you. So let's take a moment to look at this cell and talk about it a little bit and describe how this cell is going to make protein. There's kind of a big deal of proteins for cells. Most of the information inside the DNA is used to make protein. Protein is so important for your body. If you want an example of what protein looks and feels like, check out your fingernails or your hair or the rough part of your skin. That's all the protein keratin. So that's just pretty much pure keratin on your fingers there. Proteins are also really important for transport, for immunity, for making enzymatic reactions happen inside of our body. A lot of what happens in our body is done by proteins. So we want to make sure that we can make proteins, and the information we need to make proteins is stored in DNA. So our nucleus is where the DNA lives. All of this purple in here is DNA. And then there's this blue center, that's the nucleolus. That's still DNA. But the thing that makes this DNA different than that DNA is that this DNA in the nucleolus has the information you need to make ribosomes. Ribosomes are the cellular component that make proteins. So you need DNA to make a ribosome to make protein, but you need a ribosome to make the protein that the ribosome's made out of. It's kind of like a chicken or an egg situation. So all of the information you need to create ribosomes is stored together here. The reason it's stored together is because it's all used pretty regularly inside of your cell. So if you have a spice rack at home, the spices that you use most often are probably in the front all together because you need to access them quickly and easily. That's the same thing with your DNA used to make a ribosome. You put it all together so you can access it quickly and easily, and that's the nucleolus. So this other DNA is still used to make proteins, just not ribosomes. On the outside of your nucleus, you have the nuclear envelope. That's made of a phospholipid bilayer, and it has these protein channels in it. We call those nuclear pores. Those nuclear pores are where little cellular components can get in and out of the nucleus to access the DNA. The DNA is too big to ever leave the nucleus. So the DNA stays inside the nucleus and other components go in and out through these nuclear pores. Then here you can see that your nuclear envelope is connected to the endoplasmic reticulum. So the membrane that makes all of these endomembrane systems is kind of continuous, which is really cool. And because it's a fluid mosaic, it can swap parts around and be created and removed. It's very dynamic. I know that this is the rough endoplasmic reticulum, sorry, because it's studded with these little orange or red dots all over the outside of it. Those are ribosomes. We have some over here too. Those are free ribosomes. So free ribosomes floating in the cytoplasm, or ribosomes attached to the endoplasmic reticulum. So outside of your nucleus, a whole lot of endoplasmic reticulum. There's a whole lot of it because it's really important. Your cell uses it a lot. Then this structure over here that kind of looks like coral, to me it looks like coral. That is the smooth endoplasmic reticulum. It's smooth because it does not have ribosomes. And you might get it confused on some diagrams with this, the Golgi apparatus. It also does not have ribosomes, but the Golgi apparatus looks kind of like pita stacks to me, and the smooth endoplasmic reticulum looks a little bit thinner and narrower and more woven together, more network-like. That's what reticulum means, by the way, a network. Then we've got our cute little mitochondria. When we show a cell, we often show just a few mitochondria. In reality, there are thousands of mitochondria in every single one of your cells because the product that they make, ATP, is so important. Your cells need a constant supply of it, and they use a lot of it. So thousands of mitochondria in your cells, but we show just a couple for clarity and for ease. Then we have some vesicles. Over here, all this yellow, that is the cytoplasm. The cytoplasm we used to think was just like a gel that sat inside of the cell. Turns out that it's way more complex. It's actually got all of these different hormones and enzymes, and there's even concentration gradients. So like the solutes in one part of the cell would be totally different than the solutes in another part of the cell. So it's way more complex and dynamic than we originally thought. That's also true for the cytoskeleton. That's these little rays that they're showing. The cytoskeleton we used to think was just there, like a lattice or like a scaffolding that was holding up a cell. But now we know that it's actually a little highway inside the cell, and it creates itself and destroys itself simultaneously. I think of it like the staircases in Harry Potter, where the staircases are constantly moving and rearranging. That's what the cytoskeleton's like. It's changing and moving as your cell adapts to its environment. And then these little centrioles right here, those are going to be used in cell division, which we'll talk about at the end of the term. So we're going to kind of, not give them a lot of attention right now. But let's go back to our ultimate question. Our question was, how does a cell make a protein? We know that proteins are one of our 4 biological molecules. We know that they're the most structurally variable. They can take on lots of different structures, and we know that they have the most diverse functions. So they can do a bunch of different things inside of our cells, and that's why they're so important to us. And there's 2 answers to how a cell makes a protein. The first answer is, if your protein is going to get used in the cytoplasm, it's a little bit simpler. It goes like this. The information that you need to make a protein is stored in the DNA. The DNA cannot leave the nucleus. So the bit of DNA that is used to make a protein is called a gene. You find the gene that you want, the gene for the correct protein, and then you read that gene and you make it into a molecule that can leave the nucleus. We call that molecule messenger RNA. So messenger RNA copies the information in your DNA, in your gene, and then takes that message out the nuclear pore over to a free-floating ribosome like these right here. Your ribosome will read the mRNA. The mRNA tells your ribosome what order to put the amino acids in, creating your protein. The ribosome will take an amino acid, take another amino acid, and pop them together with a peptide bond, forming a polypeptide. That polypeptide will fold up into its functional shape, its tertiary shape, its 3D shape, and then it's ready to rock. You've got a protein. Fabulous. What if, though, you wanted to make a protein that was going to get ejected outside of the cell? Or if you want to make a protein and then give it to, let's say, this lysosome over here. In that case, you take a different path. You still start with the DNA. You find the gene that you want that codes for the protein that you're going to use. You make a copy of that gene in the form of messenger RNA, mRNA. mRNA can leave the nucleus through a nuclear pore, but maybe that nuclear pore just fits it right into the rough endoplasmic reticulum. So if you're going to make a protein that doesn't go to the cytoplasm, the ribosome you're going to use is going to be on the rough endoplasmic reticulum. And when your ribosome makes a copy of the message for the protein, when it makes its polypeptide, it's going to make it into the rough endoplasmic reticulum. So one of these ribosomes is going to read the mRNA. It's going to take the amino acids, put them in the right sequence. You're going to create a polypeptide, and then that's great. When you were doing it in the cytoplasm, you were pretty much done. But in this case, you're going to leave it inside of the rough endoplasmic reticulum. A piece of the rough endoplasmic reticulum is going to bud off. It's going to fuse with the Golgi apparatus. So a little piece is going to create a little transport bubble, which is going to come over to the Golgi apparatus, fuse with it. Then the Golgi apparatus is going to modify the protein. It's going to make sure it's in the right shape. It's going to take out parts, maybe. It's going to edit it. It's going to repackage it. And then it's going to bud off the Golgi apparatus, and it's going to go on its little way. So here is the protein I made in a vesicle. Maybe the vesicle will fuse with the cell membrane and it'll eject the protein. That's how you get all the proteins that you use in your saliva, is through this process, being ejected out of the cell through the vesicle. Or this vesicle could float down here and join with this lysosome. Maybe you're creating more enzymes to break open and recycle cellular components. So those are the two pathways for protein: DNA, mRNA, a free ribosome, or DNA, a ribosome on the rough endoplasmic reticulum, Golgi apparatus, vesicle, and then destination, wherever your destination is. I know that was an awful lot, very complicated, so let's look at another schematic. This is a close-up of two parts of the cell that you're going to use to make a protein. What's missing is the nucleus. Let's pretend that the nucleus is over here. I can even draw it. Yeah, so here's my nucleus. Yay. Inside I've got DNA. Okay, that's the D. This is an N. DNA inside my nucleus here. Woo! So excited with that A. So now I've got my DNA inside my nucleus. I'm going to find the gene that has the information I need for the protein I'm going to make. Let's pretend that this protein is called amylase. Amylase is used to break down starch, so you use amylase in your saliva. So I found the gene for making the amylase protein. I'm going to make a copy of that in mRNA, because mRNA can leave the nucleus. It will go over to one of these ribosomes, these red dots on my rough endoplasmic reticulum. One of these ribosomes is going to take the information in the mRNA and create a polypeptide from that information. So I've got my polypeptide now. I can take my polypeptide, put it in one of these vesicles. This little vesicle will move over. It will fuse to my Golgi apparatus. The polypeptide will be modified and repackaged. And since this is an amylase, I know that I want it in my saliva. So now I have to get it out of my cell. It's going to get packaged and shipped, and then it's going to get booted out in another little vesicle, and these bud off. So let's bud it off of the Golgi apparatus. It's going to move toward the plasma membrane. I've got my amylase enzyme inside, and it will then fuse with the plasma membrane, and it will dump the contents outside of the cell. So now I can use these amylase enzymes to break down the foods that I just ate. Cool. Now let's make another protein. let's say that we're going to make an enzyme that's used inside the lysosome. It's still not free in the cytoplasm, so I have to go the route of Golgi and rough endoplasmic reticulum. So I find the gene that I need to make the enzyme. I create the polypeptide at one of the ribosomes on the rough endoplasmic reticulum. I ship it to the Golgi. The Golgi modifies and packages it, and then it buds off. And when it buds off, it could go fuse to an existing lysosome, or it could just become a lysosome on its own. Because a lysosome is essentially a vesicle filled with hydrolytic enzymes, which are enzymes that can break down food or damaged organelles. Like if one of your mitochondria stopped working, it could fuse to the lysosome and that would destroy it and recycle its components. If I wanted to take one of my enzymes somewhere else inside the cell, so not ejected and not into a lysosome, it would bud off into a little vacuole or a little vesicle, and it would float away and take it to another component of my cell. If this were a plant cell, this little vacuole would fuse with the main central vacuole, and I would use it to make sure that all the water I'm storing inside my central vacuole is appropriately pH'd. If I wanted to make a protein that was just building in the cytoplasm, I could skip all this, and I would just go DNA, mRNA, free ribosome in the cytoplasm, create a polypeptide, and it folds up, and then it starts working. I hope that helped help you understand the process, and I want to leave you on a positive note to think about. So don't forget that whenever you feel sad, just remember there are billions of cells in your body, and all they care about is you. Thank you for watching, and I will talk to you soon.